So good morning everybody. Before I start today's topics, I like to tell all of you, while evaluating the answer script, it seemed to me that as if some of you have never appeared examination, because you have not written which question's answer is you are attempting. So take care of that. In final, they will not search which part of the question you have answered. They will just skip it. They then won't give me any mark. So when you write answer to the question number 1, say A. Next, if you are answering the question number B, then same thing you write, answer to the question number 1B. Like that, no? Specify, write clearly so that examiner do not have to spend waste his time unnecessarily. Okay, we'll start today regulation of gene expression. Time is very short, so we have to go a bit fast. Probably you will not have class from 25th of July, no? You have requested, so we are trying to finish up as early as possible. Now, this is a very important topic. You will get long answer type questions about regulation of gene expression in eukaryote. They may specify as eukaryote, or regulation of gene expression in prokaryote, or write a note on Lac Operon. So let's see. Now, synthesis of protein under influence of gene is called gene expression. In molecular biology, the central dogma is like that only. From DNA to protein synthesis, in between from DNA there is synthesis of RNA, and there is translation into protein. Now, DNA is the chemical vehicle of heredity. Its functional unit is gene. Now, every organism has so many genes, so many information in DNA that collectively forms the genome of the organism. That means the term genome refers to the total genetic information contained in the cell. Now, bacteria E. coli, having at least approximately 4,400 genes, and human being is having almost 19,000 to 20,000 number of genes. You know, all genes, they are present in every tissue, but through this regulation of gene expression, some particular gene is selectively expressed in the selected cell. That is the function of regulation of gene expression. Now, at any given time, only a fraction of the genome is expressed. The living cell possesses a remarkable property to adapt to changes in the environment by regulating the gene expression. Now, to survive, the organism has to adjust with the environment, and that needs functioning of different proteins, different enzymes for adaptation, and that is done by the gene only. The gene is expressed according to the need of the organism. Insulin synthesis is occurring in the beta cells of the pancreas, but insulin gene is present everywhere. It is present everywhere, but they are not expressed, they are regulated to be expressed in beta cells of the pancreas. Now, some of the genes, some regulatory element, they increase the expression of gene in some given situation. Some elements, they decrease the gene expression. So, when it is increased, it is called positive regulation. When it is decreased, it is called negative regulation. See, these terms in genetic, the terms are very important. Okay, inhibition, we tell it as repression in case of molecular biology. So these terms you have to remember. So positive regulation and negative regulation, negative regulation we could have told it inhibition, no? But in molecular biology it is told as negative regulation. See, all cells need glucose for their metabolism. So, if in a given time, if the citric acid cycle enzyme is stopped synthesizing, what will happen to the cell? It will die. There will be no energy generation. Every minute, every second, energy is required. So, this type of gene, they are called housekeeping gene. That means they are not regulated. Every time they are expressed, every time they are synthesizing the enzyme in all cells. So, these are called housekeeping genes or constitutive genes. The product of these genes are required all the time in the cell. Therefore, the constitutive genes are expressed at more or less constant rate in the almost all cell. They are not subjected to regulation. Enzyme of citric acid cycle, one of glycolysis. Okay, these are always synthesized. Inducible genes, here the protein part may not be required every time, but in a given situation, there should be synthesis of that protein. So, there is a regulatory mechanism. When we don't need, this will be this protein will not be synthesized, and when we need it, the protein will be synthesized. So there is a regulatory mechanism. So this type of gene are called inducible gene. The concentration of the proteins synthesis by inducible gene is regulated by various molecular signals. An inducer increases while repressor decreases. Example one, tryptophan pyrrolase in the liver. It is induced by tryptophan. Pseudogenes, what are these? These are the genes having same structural homology with an active gene, but these are not expressed because of the mutation. However, they are carried down to the offspring as hereditary information. They will be carried to the offspring, but they are not expressed due to the mutation. Previously, as the enzyme is a protein, take an example, enzyme. It is a protein, so it will be naturally coded by some gene. So previously thought that for each enzyme, there is a particular gene. Okay, so that time it was told that one gene, one enzyme. But it has been, we have seen that some enzymes are there where there is 2-3 subunits are there, and these subunits may not be the identical one. They are synthesized by the different fraction of DNA. So, the portion of the subunit which is synthesized is called cistron. So, nowadays it is said one cistron, one subunit. The general concept of one gene, one enzyme is replaced by one cistron, one subunit. Gene expression in prokaryote. Now, to explain the gene expression in prokaryote, they have adopted a model of Lac Operon. This model is based on regulation of lactose metabolism in E. coli. The operon is the coordinated unit of genetic expression in bacteria. Now, here is a structure of Lac Operon. The Lac Operon, this is the Lac Operon gene. It has a regulatory gene here, I. Regulatory gene I. It inhibits the rest of the expression of the rest of the structural gene. It inhibits. Its function is to inhibit. So that's why it is given as I. So regulatory gene I, inhibition is function. Then this gene has a operator gene here. This operator gene, it functions as the expression of the gene XYA, and a promoter region here in between the inhibitor and the operator gene. ZYA, not XY, ZYA is the structural gene that is responsible for synthesis of enzyme. Okay? Now, gene Z, it synthesizes beta galactosidase. Gene Y synthesizes galactoside permease. And gene A synthesizes acetylase. Now see, here in this structural gene, three components are there which synthesize three different enzymes. But when mRNA is synthesized, the whole mRNA containing the different translational unit are synthesized. Such type of mRNA is called polycistronic mRNA. I told you this part is considered as cistron, responsible for the synthesis of beta galactoside. This part is cistron, responsible for galactoside permease, and next is acetylase. So that's why it is called polycistronic mRNA. In prokaryote, this whole thing is synthesized as one mRNA, and subsequently the required protein is synthesized from this mRNA. But in eukaryote, it is not like that. In eukaryotic, regulation of gene is very complicated and different. Now, depression of Lac Operon in absence of lactose. This regulatory gene I, it synthesizes Lac repressor, another protein, Lac repressor. This is a tetrameric protein. It is a regulatory protein. This protein, repressor tetramer, it binds with the operator gene specifically and prevents binding of RNA polymerase to promoter site. Here is the binding site for the RNA polymerase. So when this repressor tetramer is bound to the operator gene, RNA polymerase cannot bind with the promoter site. So there is no synthesis of gene mRNA, the polycistronic mRNA. So there is no production of those three enzymes. So it does negative regulation of the gene expression in absence of lactose. There is no lactose, so there is repressor tetramer is synthesized and it binds to the operator gene, inhibiting the binding of RNA polymerase to the promoter site, and thus the negative regulation of the gene is done. It is in absence of lactose. Now, when there is presence of lactose, when lactose is present, what it does? Some amount of lactose it diffuses inside the bacteria and it binds with that tetramer and causes conformational change. So, some amount of lactose enters in the E. coli and binds with the repressor molecule due to the high affinity. That lactose has high affinity towards the repressor molecule, so it binds. Simply, it binds on the repressor molecule. That causes conformational change in the lactose repressor molecule. That due to that conformational change, the repressor becomes inactive and cannot bind to the operator site. So it cannot bind to the operator site, thus leaving the promoter site suitable for binding of RNA polymerase. So RNA polymerase will now bind in the promoter site and there will be synthesis of that polycystonic mRNA. So lactose acts as inducer by activating repressor and there will be utilization of lactose by the E. coli. Now, when there is both the glucose and lactose is there in solution and E. coli is there in solution, what happens? The E. coli prefers utilizing the glucose because it is not that E. coli likes glucose very much, because glucose has a inhibitory action on Lac Operon. Okay. Now how it does? Some amount of glucose it diffuses inside the bacterial cell and it inhibits adenylyl cyclase. To bind the RNA polymerase in the promoter site, it requires a protein called cap bound to cyclic AMP. So here the glucose it inhibits the adenylate cyclase and there is depletion of cyclic AMP. So once there is depletion of cyclic AMP, there is not enough cap cyclic AMP bound molecule. So that one RNA polymerase cannot bind to the promoter site and there is no polycystonic mRNA. So lactose utilization is inhibited by the glucose. But as soon as utilization of glucose is over, then the inhibition of adenylyl cyclase is removed and there is again synthesis of polycystonic mRNA. So again it will use lactose. So these are the regulation of gene expression in prokaryote. Now gene expression in eukaryote, it is a very complex process. Each cell of the higher organism contains entire genome. Eukaryote gene expression is highly regulated and provide the appropriate response to the biological need. It may occur expression of certain gene or activation of selected gene upon demand or permanent inactivation of several genes in all but a few types. In case of prokaryotic cell, most of the DNA is organized into gene which can be. In contrast, in mammals, very little of the total DNA is organized into gene and their associated regulatory sequence. However, a lot of DNA is there inside the body. The non-gene part of the DNA and its function is not known. The function of the bulk of the extra DNA is not known. Now, eukaryotic gene expression and its regulation are highly complex, and some important steps we will discuss. Everything we won't be able to discuss. So, now, here is the DNA. The first RNA which is synthesized is called heteronuclear RNA or primary transcript. This primary transcript is modified to mature mRNA. Then only it goes for protein translation. So here, in the gene, in the chromosome, gene is not just exposed to for regulation. It is hidden in multiple turns, then foldings of the chromosome. So, before DNA, the genome, this chromosome has to be modified so that its gene can be approached for expression. So here, one step is epigenetic. So, in epigenetic modification, the chromosome will be or gene will be modified in such a way that there is expression of the gene becomes easily approachable for expression. Now, it happens in the chromatin structure and there is gene expression. How it happens? Two things happens. There is histone acetylation and deacetylation. In the chromosome, the DNA, it is wound around histone protein, forming a nucleosome. So it is not readily available for, this is histone octamer. Around that histone octamer, this DNA is wound around. Okay. Now, to make it available for expression, there should be looseness or it should be unwound from the histone so that it can be approached. So, DNA in hydrogenism packed as protein DNA complex called chromatin provides additional level of control of gene expression. That means gene is not readily available for expression. So, genes that are transcribed in particular cell are less condensed and more open. Now, it has to be less condensed and more open type. So, how it happens? There is acetylation of lysine residue in amino terminal end of the histone. When there is acetylation, there is decrease in the positive charge of histone. Now DNA is negative charge. Histone now has become less positive charge, so there is less affinity of histone towards DNA. So there will be loosening of the DNA from histone, and that favors genetic expression. Now, thus binding ability to negatively charged DNA is reduced, and that will cause disruption of nucleosome structure to allow transcription. You have understood this part? When you are going to write in the as answer, no? You don't have to write so many things what I have told. What is gene expression? Regulation of gene expression, you just tell. What is the need of gene expression and levels of regulation of gene expression. Now in case of epigenic gene expression, this histone acetylation and histone methylation is important. These two you have to describe. And you don't have to go so many things. In book, so many things are there. So many things you will not be able to write in exam answer sheet. So write the important things. Okay? So in case of, in the interest of gene expression, that DNA protein complex has to be loosened and DNA gene should be available for expression. How it is done? That is done by acetylation of the lysine residue of amino acid terminal end of the histone. That causes decrease in positive charge of the histone and affinity towards the DNA is reduced. So there is disruption of the nucleosome structure and it allows transcription of mRNA from the gene of interest. Now what happens in methylation? Methylation occurs in the cytosine residue in the CG sequence of DNA to form 5' methyl cytosine, which causes loss of transcriptional activity due to binding of methyl cytosine binding protein. Methylated DNA does not expose for binding of transcription. Methyl cytosine binding protein, it will bind to the methylated site. Here is the DNA binding protein. Here is the polymerase II binding site. So if it is not methylated, the DNA binding protein can bind here and gene activation and expression starts. But in case of histone methylation, the methylated transcription protein, it binds to that methylated portion of the DNA. And when there is binding, that protein doesn't allow binding of the DNA transcription factor. That's why there will not be any transcription. So methylation causes inhibition of the transcription. So in epigenetic modification, these two things we will discuss. We will write. Take it from here. I have just concised it. You will have it will be helpful for you. So here it is methylated. Methyl binding cytosine binding protein is attached here. So there is this part. That means this polymerase binding site is not free for binding of the polymerase DNA binding protein. So there will not be any transcription. Now enhancers and tissue specific gene expression. This will go a little bit later. Come to the now in gene level, there is gene amplification and gene rearrangement. These are in the gene level. So, epigenic level, we have exposed the gene for expression, and if there is methylation, there is gene is not available for expression. Okay, so this acetylation and methylation, how it causes influence gene expression, you have known the thing. Now, in case of gene level, we have enhancers, transcription factor, we have gene amplification, we have gene rearrangement. Not what you are discussing, huh? So, gene amplification. What happens? Here is a gene. Suppose here is a gene. For example, what has been described here as a methotrexate related gene, dihydrofolate reactive gene. Dihydrofolate reactive gene this gene, dihydrofolate reactase, it synthesizes dihydrofolate and tetrahydrofolate, responsible for DNA synthesis. Now, in case of cancer, there is huge utilization of these nucleotides because cancer cell is multiplying like anything. Okay. Now, we give a anti-cancer drug, methotrexate. What methotrexate does? It inhibits synthesis of dihydrofolate and tetrahydrofolate. Thus, DNA synthesis is inhibited. So, there is no growth of cancer tissue. Okay. Now, when this drug is given for prolonged time, what happens? There is a phenomenon called gene amplification. The genes are multiple copies of genes will be created so that many and these genes will be available for synthesis of dihydrofolate. So many genes will be there for synthesis of dihydrofolate. Okay. Now, once drug is given, it will inhibit some few numbers of genes, but there will be so many genes which will not be inhibited by the methotrexate and will function as dihydrofolate reductase synthesis. Okay. So, thus the methotrexate become resistant. Okay. And this is done by gene amplification. Now, in case of the next one is gene rearrangement. What happens? So for example, this one is a immunoglobulin gene. It has short immunoglobulin light chain. So this is the variable region, junctional region, constant region. So there is about 500 number of genes in the variable region. There is 6 numbers of genes in this joining region, and constant region has about 20 number of genes. So some number of variable gene will be picked up with the junctional, gene of the junctional gene, and some from the constant region gene. So here it has selected. There is a rearrangement of DNA has been done here. Then primary transcript is synthesized, hnRNA, heteronucleic RNA. Then from there it has clipped off this part and kept these 3, and from there the protein of light chain is synthesized. So now you imagine from 500 number of light chain variable gene and 6 number of this joining gene, 20 numbers of constant gene. If they in combination and permutation and combination, how much number of immunoglobulin, different type of immunoglobulins will be synthesized. Okay, thus there is a great number of antibody diversity in this way, by means of gene rearrangement. Okay, understood this part? This part you have to explain. So this is gene amplification. Here gene is amplified in multiple number. Here gene is rearranged to produce diversity. So these are the in genetic level. Now combination of DNA elements and protein in gene expression. Enhancers and tissue specific gene expression. The DNA elements that enhances gene expression by providing binding site for the protein that regulates transcription. There are DNA sequence, some sequence, they allow binding of the enhancers and initiate synthesis of the RNA. One example, that steroid hormone receptor binding complex that binds with the element in the DNA and produces the required protein. In case of vitamin D, it is calcium binding protein in the intestine. No? So that is the thing, it is like said here. So there is a DNA element that enhances gene expression, providing binding site for the protein that regulates transcription. Facilitated binding of the transcription complex to promoter region. Tissue specific gene expression is largely mediated by the enhancers, examining immunoglobulin gene in beta lymphocytes, lymphoid tissue. Now combination of DNA elements and protein in gene expression. In this example, this gene 1 is activated by protein 1, 2 and 3. Here gene 2 is activated by protein 1, 3 and 4. Here one thing you note that protein 1 and 3, it has directly bound to the gene, but the 4 is bound to the protein 1 and 3. So for influencing the gene expression, the protein may not bind to the gene directly, but can activate by binding to the other proteins that is bound to the gene. Here in gene 3, another protein 5 has bound to the gene, which inhibited the binding of protein 2. So the gene inactivation has been done by the 5. So the protein 1, 2 and 3, which is required for binding of for expression of the gene 1, is inhibited by the protein 5, due to binding of which there is this protein 2 cannot bind to the protein and initiate gene expression. So this is combination of DNA element and protein in gene expression. Now motifs. Motifs, we have known that 4 levels of protein structure. This is the super secondary structure. This is a different structure. This is super secondary structure of protein that influences the gene expression. Now motifs are literally mean dominant element. Certain motifs protein radiate binding to transcription factor. Specific control transcription occurs binding to the regulatory protein with high affinity in the correct region of the DNA. These motifs, they bind to the major or minor groups of the DNA, and binding of this motif will help binding of the transcription factor in the gene. 4 unique motifs are here: helix turn helix motif, zinc finger motif, leucine zipper motif, helix loop helix motif. Now if you in detail if you go, there is a thick book, so that is not possible to write in the exam. So just few words you can mention. In case of helix turn helix motif, it is about 20 amino acid component of protein. Example: lactose repressor, cyclic AMP catabolite activator protein. Few slides back I have told you. Zinc finger motif. Zinc finger motif, here zinc is bound to the cysteine residue or histidine residue and make a finger-like structure. That finger-like structure, it binds to the major group or minor group of the DNA and helps transcription. Helps in transcription. Example: steroid hormone receptor transcription factor. Leucine zipper motif. See, leucine zipper motif. Here 2 polypeptide chains are there. This polypeptide chain has leucine. They bind together to like a zipper. Okay, like a zipper here. So that amino terminal end. This is the amino terminal end of the zipper. It binds with the DNA and facilitated transcription. Okay, Phos and Jun are proteins, transcription protein. They bind with leucine zipper motif. Helix loop helix motif. Here 2 alpha helical structure that binds to the DNA. Here helix turn helix motif. So we have described gene level, gene amplification, gene rearrangement, then those enhancers and motifs, how they bind. Now after transcription, our mRNA is synthesized, means hnRNA is synthesized. Now hnRNA will go different type of processing to form mature RNA. These are alternate processing of mRNA, alternate mRNA splicing, transport of mRNA to cytoplasm from nucleus, then degradation of RNA. So now processing of RNA. Processing of RNA, the whole RNA is not required for gene expression, or it doesn't come under the gene. Okay, now suppose this part of our mRNA So this is axon, intron. So here intron axon splicing will be there. The intron, this will be removed and axons will be joined. Thus, non-coding sequence will be removed and the coding sequence will be joined together. That is called RNA splicing. Now, it has become a mature RNA and ready for transcription, translation. Now, alternate mRNA splicing. Here, what happens? Alternate mRNA splicing means there will be so many exons will be there. So required exon will be spliced, joined together. So in between, like intron, the exon part which is not required for the genetic expression will be removed. Degradation of mRNA. Degradation of mRNA. If mRNA is degraded very fast, there will be no enough time for synthesis of enough protein. So mRNA will be stabilized so that it can last for a long time and synthesize protein in adequate amount. Now here estradiol, this prolongs the half-life of vitellogenin mRNA from a few hours to about 200 hours, so that enough protein can be synthesized. So these are the modifications that occurs in the mRNA. Epigenetic therapy of cancer. Now, when cancer, what is it? There is a enormous cell growth. Gene is involved in this. So we have known that methylation can cause gene, can inhibit gene expression, so we can take that advantage. No? Here you see tumor suppressor gene. Now, tumor suppressor gene, when it is expressed, it prevents formation of tumor. Tumor suppressor gene, it will suppress formation of tumor. Okay. Now, now it has seen that this tumor suppressor gene has multiple methylation in the gene part, and DNA methyl transferase is responsible for that. Okay. So what we do, we give a molecule which inhibit this DNA transferase enzyme, DNA methyl transferase enzyme. So when this DNA methyl transferase enzyme is inhibited, there will not be any methylation of tumor suppressor gene. So tumor suppressor gene will be expressed and thereby inhibiting the generation of tumor. Okay. Histone deacetylase is another enzyme. This enzyme stimulates tumor suppressor gene expression. We told histone when it is acetylated, it opens the gene for expression. So we will allow this tumor suppressor gene to get expressed by enhancing the acetylation of histone, thereby increasing the tumor suppressor gene, and there is prevention of cancer. So this is this 5-azacitidine. This is already FDA approved. It is used in the treatment of leukemia, and verinostate is used in T-cell lymphoma. So epigenic therapy of cancer by employing various inhibitors individually or in combination holds a great promise. Okay, so it is very important chapter. Don't miss it. You may get a long question. Okay, thank you.